Imagine a world plunged into darkness. It wiped out over 50 million people in the 14th century. Entire cities emptied, societies collapsed, and panic spread faster than the disease itself. But did you know the same bacteria still exists today? This is the story of Yersinia pestis, the bacterium behind the plague. Let's explore its biology, history, how it spreads, and why it still matters today. Imagine a world plunged into darkness. It is the middle of the 14th century. Fear walks the streets of every town and village across Europe. A mysterious sickness arrives, silent and swift. People fall ill with strange, painful swellings on their bodies. Within days, many are gone. This terrifying illness was called the Great Pestilence, or the Black Death. It was a shadow that fell over the land, changing everything it touched. They did not know that the true killer was something incredibly small. It was a living organism, a microscopic hunter. This tiny agent of death was a bacterium. We now know its name, Yersinia pestis. It is a name that carries the weight of history's greatest tragedies. The story of this bacterium is a powerful reminder of the delicate balance of life on our planet. The plague did not disappear after the Middle Ages. It is still with us today. Understanding this tiny organism is not just about looking into the past, it is about protecting our future. The tale of Yersinia pestis is a dramatic story of survival, both its own and ours. So what is this microscopic creature that brought empires to their knees? Yersinia pestis is a type of bacterium. Bacteria are single-celled organisms among the most ancient forms of life on Earth. They are everywhere, in the soil, in the water, and even inside our own bodies. Most are harmless and many are even helpful. But some, like Yersinia pestis, can be incredibly dangerous when they enter a host that is not adapted to them. When they enter a human, the results can be catastrophic. This particular bacterium is rod-shaped, a simple form for such a complex and deadly organism. Its discovery is a more recent chapter in our long history with the disease. In 1894, Alexander Yersin traveled to Hong Kong where the plague was raging. Working in a small hut, Yersin took samples from the buboes of victims. Using a microscope, he was the first to see and identify the tiny, rod-shaped bacterium responsible for the disease. It was a monumental discovery that finally gave a face to the ancient enemy. This tiny villain had evolved remarkable ways to survive and cause disease. It produces proteins that act like a shield, making it almost invisible to our immune system. Our body's defenders cannot easily recognize or destroy it. This allows the bacteria to multiply very, very quickly. Scientists believe Y. pestis evolved from a much less harmful gut bacterium through a few small genetic changes. It gained the ability to survive in fleas. This was the key that unlocked its potential for devastation. By living in fleas, the bacterium found a perfect vehicle to travel from animal and to humans. This evolutionary leap turned a relatively minor germ into a world-changing killer. It shows how small changes in nature can have enormous consequences. Next, how the plague travels. A chain of life that links fleas, rodents, and humans. The spread of plague is a dramatic story of interconnected life, a chain of transmission that links the smallest insect to the largest cities. The main characters in this journey are rodents, rats, mice, squirrels, prairie dogs. In many parts of the world, Y. pestis lives quietly within wild rodent populations. These animals are its natural reservoir. When a flea bites an infected rodent, it sucks up blood filled with Y. pestis. Inside the flea's gut, the bacteria multiply and form a sticky biofilm that blocks the stomach. The blocked flea becomes hungry and bites again, regurgitating bacteria into the new host. Historically, rats traveled on ships carrying infected fleas across oceans and continents. When rats died, fleas abandoned carcasses and sought new hosts, often humans. This created the perfect storm for rapid spread. But fleas and rodents are not the only route. If someone develops pneumonic plague, they can infect others by coughing. Pneumonic transmission allows person-to-person spread without any insect or animal intermediary. Plague is not just one disease, it reveals itself in 3 different forms, each with its own set of terrifying symptoms and dangers. We will look at bubonic, septicemic, and pneumonic plague. Bubonic. This usually happens after a person is bitten by an infected flea. Bacteria travel to the nearest lymph node and multiply rapidly. The lymph node becomes swollen, painful, and hard. These are called buboes. They often appear in the groin, armpit, or neck. Without treatment, bubonic plague is very serious. Mortality historically 30–60% if untreated. Septicemic. If bacteria escape the lymph nodes, they can multiply directly in the blood. This causes sepsis, organ failure, shock, and problems with clotting. Bleeding under the skin and blackened tissue can appear, hence the name Black Death. Septicemic plague can be nearly 100% fatal if untreated. Pneumonic. This is the only form that spreads directly person to person via droplets. It destroys lung tissue, causing cough, chest pain, and bloody sputum. Symptoms appear quickly, and death can follow in a day or two without treatment. Pneumonic plague is highly contagious and highly lethal if untreated. The journey inside the body determines which form a person will have. In all cases, speed is critical. The faster someone gets antibiotics, the better the chance of survival. Each form presents different threats and requires immediate medical attention. Understanding these forms helps explain how outbreaks became so devastating in the past. Now, let's look at the great pandemics that shaped human history. History has been shaken by three enormous waves of plague known as pandemics. The first was the Plague of Justinian. It struck in the 6th century, beginning in the Byzantine Empire and Egypt. Historians estimate tens of millions died, weakening empires and changing geopolitics. It fundamentally altered the course of history in Europe and the Middle East. The second, and most infamous, was the Black Death, which arrived in Europe in 1347. It spread along trade routes carried by ships and rats. In just a few years it reached almost every corner of the continent. The impact was catastrophic. Estimates suggest 30–60% of Europe perished. Labor shortages empowered workers, accelerating the end of serfdom and reshaping economies. The trauma changed art, religion, and culture for generations. The Black Death was not a single event. After the initial wave, it returned for centuries. Smaller outbreaks kept populations in fear and recovery slow. By the 18th century, Europe began to see the plague recede. The third pandemic began in Yunnan, China, in the mid-19th century. It spread to Hong Kong and across the globe via steamships. This wave killed more than 12 million people, mostly in India and China. But it also saw scientific advances. Yersin and others identified the bacterium and its vectors. Understanding rats and fleas changed the battle against the disease. Public health measures and treatments began to limit plague's impact. Next, plague in the modern world. The pandemics left deep scars and reshaped societies, politics, and culture. Their echoes are still visible today. Understanding past pandemics helps us prepare for the present. It may be surprising to learn that the plague has not been completely wiped out. Yersinia pestis still exists today, though it is rare. It survives in wild rodent reservoirs. Small numbers of human cases are reported each year, mainly in the DRC, Madagascar, Peru, and parts of the U.S. Democratic Republic of Congo. Madagascar and Peru are hotspots. In the United States, occasional cases occur in rural western states. Arizona, Colorado, and New Mexico report the most U.S. cases. Plague circulates in animals like prairie dogs, squirrels, and wood rats. Monitoring helps prevent animal-to-human spillover. The good news, we have effective antibiotics: streptomycin, gentamicin, doxycycline, ciprofloxacin. Early diagnosis and prompt antibiotic treatment greatly reduce mortality. Thanks to modern medicine, death rates today are low, around 10% with treatment. Delay in treatment, especially for septicemic or pneumonic forms, can be deadly. Because we know how it spreads, prevention is straightforward. Control rodent populations and clear brush and rubbish where rodents nest. Use flea control on pets if you live in or visit endemic areas. In outbreaks, rapid public health response, contact tracing, prophylactic antibiotics, stop spread. Modern diagnostics let labs identify Y. pestis quickly. Prepared public health systems and antibiotics are powerful defenses. With knowledge, medicine, and surveillance, plague is a manageable threat today. Next, how doctors and labs unmask the killer. Fast, accurate lab work saves lives. When a doctor suspects plague, it is crucial to confirm the diagnosis quickly and accurately. They will draw fluid from a bubo or take blood or respiratory samples as appropriate. Samples are handled with extreme care and sent to specialized public health labs. Under a microscope, Y. pestis often shows bipolar staining that looks like a safety pin. Classic microbiology grows the bacteria on agar to confirm living organisms. Growing colonies give strong evidence of active infection. Modern molecular tests like PCR detect the bacterium's DNA within hours. These sensitive tests let clinicians begin targeted antibiotic treatment quickly. Rapid diagnosis and treatment can be the difference between life and death. Plague is a notifiable disease in most countries. Cases trigger public health action. Tracing and monitoring contacts and giving prophylactic antibiotics when needed prevents spread. Accurate lab work and swift public health response keep communities safe. Because Yersinia pestis is so dangerous and has caused terror throughout history, some have considered using it as a weapon. It is classified by CDC as a Category A agent. Pneumonic plague could be aerosolized. The terrifying scenario, aerosolized pneumonic plague causing rapid person-to-person spread, but weaponizing plague is difficult, dangerous, and illegal, and modern public health systems are designed to detect and respond quickly. International laws forbid biological weapons, and surveillance plus antibiotics form our defense. The story of Yersinia pestis is far more than a frightening tale from history books. It is a living lesson about how microbes can still affect our world, especially with climate change and antibiotic resistance. Tracking reservoirs and investing in public health keeps us safe. In a connected world, the lessons of plague are more relevant than ever. It teaches humility and the need for scientific vigilance. Investment in science and public health is our best safeguard. Yersinia pestis may no longer kill millions, but it's far from extinct. Its history is a lesson in how tiny microbes can reshape entire civilizations. From mystery to identification to treatment, science turned the tide. Preventive steps are simple. Avoid contact with wild rodents, control fleas on pets, and keep yards clear. Public health surveillance and early treatment stop outbreaks. Early antibiotics make plague a treatable illness today. Remember, know the risks, follow guidance, and seek care early if you are ill. If you found this video fascinating, like and subscribe for more deep dives into the hidden world of infectious diseases. The tale of the plague is both a warning and a story of how human ingenuity can overcome fear. And remember, wash your hands and watch out for fleas. Thank you for watching.